It's a great privilege to be here, and thank you so much for inviting me with these amazing speakers here. And thank you for the bravery of organizing a physics workshop in this conference. So what I'm going to talk about is first of all, my talk is a bit different from what appears in your. I realized it was a bit ambitious with the original plan. So we're just going to talk about differentiation and reprogramming, and we're going to try and give a bit of a new perspective on this question of how the cells. So we know that the different cells in our bodies, they share the genome, but they're sort of like grouped into different cell types with distinct expression profiles. So the question that we're going to try to address today, again, from the point of view of simple physics-style modeling, is the question of how the cells acquire and maintain these stable identity patterns and how they move between different identity patterns. So there are generally two ways that we tend to distinguish. So the first, and this is sort of like illustrated by this famous Waddington landscape, is this idea of hierarchical differentiation. So the famous system that we look about a lot is the blood, right, where we have this rather homogeneous population, stem population, that can establish the full range of different types of cells that are in the blood through this hierarchical differentiation program, okay? So where the population is initially multipotent, and as differentiation progresses, the cells move into more and more restricted progenitors that can generate limited sets of blood cells. And this system has been studied in many different ways by many different labs, I think, in the past, at least 50 years. Now, one of the hallmarks of this hierarchical differentiation is this idea of multilineage priming, right? So we have our terminal cells, and each of them has some transcriptional profile, which is associated with distinct sets of transcription factors that are activated, and that we know that the progenitors, they tend to co-express different programs of their target cell states. Okay, so this has been established in hematopoiesis, but also in other, let's say, adult homeostasis like the intestine, and also in developmental context, okay? So this idea of hierarchical differentiation through multilineage priming. But this is not the only way, and the other way, at least the other famous way, is reprogramming. So we can actually move directly between different cell types through the overexpression of some specific transcription factors. And now this is obviously a very big deal in aging, and this is sort of like, we have these sort of like well-established recipes. So if we take, let's say, a fibroblast and we overexpress these and these transcription factors, we can end up with this type of cell. So what we're going to try and do is we're going to try to build sort of like the simple model that can try and recapitulate sort of like these high-dimensional dynamics of these systems. And hopefully we'll also make some new predictions and think about new ways we can actually control these types of processes. So the thing is that the system, as a starting point for simple models, it's not maybe great. Because what we know is that these types of hierarchies, they tend to be established by not a small circuits of few transcription factors, but by these dense and highly interconnected networks of many different transcription factors. Now, the way this works, so the way that transcription factors regulate their own expression, it happens through binding of these cis-regulatory elements called enhancers. Now, each enhancer can bind many transcription factors, and this can cause the potentiation of expression of one or more genes associated with the enhancer. So I said each enhancer can bind multiple transcription factors. It can regulate multiple genes, right? And that what we know is that the types of interaction networks that control cell identity tend to involve, could be dozens, even hundreds of transcription factors interacting with potentially thousands of different enhancers. So what we're going to do is we take a starting point, which is the most general model that we can possibly imagine for the system, where each transcription factor can be regulated by each enhancer, okay, with some sort of a coupling rate. And each enhancer can be bound by each transcription factor. Now, we also have all the influences that come outside of this network, which we call signaling. So signaling can also perturb the different enhancers of the network. So what we're going to do is we can try and translate this sort of like network image into a dynamical system. And for this, what we're going to do is we're going to think about the sort of like basic process that actually underpins this network, which is that the binding of transcription factors to enhancers causes the recruitment of cofactors, and these cofactors modulate the chromatin of the enhancer, and thus increasing its rate of transcription initiation. And we'll also consider that the different enhancers compete with each other for recruiting this transcription initiation machinery. Okay, so if one enhancer, let's say, has more acetylated chromatin, then it has a higher rate of recruiting the transcriptional machinery and thus reducing it to the other enhancers in the system. And we can convert this into a rather simple model. So this is the high-dimensional model. So that whatever is involved is sort of like this vector matrix. Okay, so we have the dynamics of our cell identity transcriptional network, which is regulated by this coupling from the enhancers times this rate of transcription initiation by the enhancers, and that this itself has to do with the binding of the transcription factor to the enhancers plus the effect of signaling. And we also have this beta term, and this beta term corresponds, you can think of this sort of like the efficiency in modulating chromatin. Okay, so this whole network is very complicated, but what we can do is we can try and simplify it by looking at the actual experimental observations on this process. So what we know is that the enhancers that are associated with cell identity, they tend to be activated in a very cell type specific way. So only activated in maybe like a single cell type, or one or two cell types. Okay, and we also know that these transcription networks are not just random networks, but they have a very specific regularity. And that regularity is that the transcription factors that are expressed in the same cell type, they tend to all co-bind their own enhancers. So we can think of this as sort of like a clique in a way, right? So we have similar transcriptional binding profiles of these enhancers that activate these transcription factors, and they all co-bind the same enhancers. So what we can do is, first of all, with some algebra magic, we can actually say that our model doesn't capture physical enhancers, but it captures enhancer types, okay? It's about the binding profile to the enhancer. And the other thing we can show is that there's some sort of a fundamental symmetry in this network where the coupling, so the coupling of a transcription factor to the enhancer is similar to the binding of that transcription factor to the enhancer. So it's a very basic symmetry in the dynamics. So basically our Q and our Xi matrices are very similar to each other. And for the sake of this talk, we can assume they're basically the same, and it doesn't make any difference. Okay? And finally, the actual row of Xi should correspond to the expression patterns of the transcription factors in the network. Okay, so we have a much more simplified model that you can actually mostly parameterized by only looking at the terminal cell states in the system. And we can actually prove this, that when the chromatin efficiency beta is sufficiently large, then these dynamics can, in fact, stabilize all the different transcriptional profiles given by this matrix Xi. Okay, so we have this dynamical system that can actually stably maintain expression patterns. Okay, but this is not the title of the talk, right? So we want to ask this basic question of not just how they're maintained, but how the cell types actually move between these expression profiles. So the first thing that we can do is we can ask, so what happens if we do this type of reprogramming style perturbation where we overexpress specific recipes of transcription factors? And this actually selectively modulates this attractor network. So we have this type of, I think it's like a physical landscape. Okay, and what we do when we overexpress transcription factors is that we modulate this landscape so that certain patterns become much more attractive than others. They can even encapsulate the attractiveness of other patterns. But this is not just an abstract concept, right? We can actually, we have the dynamical system, which is parameterized by the terminal identities. So we can ask, so what happens if we actually do this thing, do this type of like in silico style experiment of overexpressing specific transcription factors. So here, for example, we overexpress ASCL1, and we start from a fibroblast, and we actually do end up in a neuron as we would expect. And we can do this for many different reprogramming recipes, and it works. Okay, so what this basically says is that the success of reprogramming at the end has to do with the statistical nature of the patterns that the system is storing. Okay, because we can recapitulate this with these types of simple in silico perturbations. But this is not how differentiation actually looks like, right? This is not this nice hierarchical pattern that we see, for example, in the blood. So to recapitulate that, so we need to look at the other parameter, beta, which is this chromatin efficiency parameter. So whatever we did before had to do with modulating something that is akin to W, right? Everything that's upstream of the network is W, including if it's a transcription factor overexpression. But we're going to try and ask, so what actually happens if you modulate this beta, this chromatin efficiency? So we said when this chromatin efficiency is large, then the system retrieves its own patterns, retrieves the cell types, so it selects specific enhancers. But actually, when we decrease beta, okay, what we do is we actually destabilize our attractor patterns, and we move towards these sort of like meta patterns, which are averages of our N-type pattern. And then as we increase beta again, what we do is we move always to more and more restricted patterns. And at the end, what we do is we converge to an endpoint pattern. And actually, we can show that this endpoint pattern is sensitive to the signaling of the system. So even though we didn't perturb, so we still have this complicated attractor landscape, we can move between patterns in a way that's sensitive to the induction of these patterns by signaling. And we can do this for something like a toy example. Okay, and we can see, and this is something we can also prove this mathematically, we can see that we always move in a way that looks a lot like these multi-lineage priming. We move between averages, and we move through bifurcations. But these bifurcations are not just random, right? We don't just move through any possible sort of like progenitor state. We move through specific progenitor states that we can also anticipate based on the statistical structure of our end patterns. Now, the final thing we can do is we can ask, so can we do this for something like hematopoiesis? So if we look at the terminal cell states, and we run this procedure, which we call annealing. So it's similar in a way to different things in physics called annealing. So we call this annealing procedure. So we run this annealing procedure, and we can actually reconstruct something that looks a lot like this canonical hematopoiesis tree. I mean, it looks a lot like the types of things that people actually see in, for example, single-cell experiments, including, by the way, these types of like alternative differentiation pathways. And we recapitulate this multi-lineage priming in the progenitor states. But the important thing is we didn't have basically any fitting parameters here. This entire tree, in a way, emerges from what we think of as a sort of like a search process, okay? So we think about this something like a hematopoietic or a hematopoietic stem cell that needs to be stable for long periods of time, but then occasionally has to move and become something like a red blood cell. Okay, so we need to have these two things as stable for long periods of time. And the way the system does this, that we think about, is what it does, it moves in this type of search process, or this annealing process, where it can converge to an induced cell type. But what we see when we see this progenitor tree is we see something that's basically the byproduct of this search process. Yes, so just to sum this up, so this is obviously, it's a ridiculously simple model. But hopefully there is something behind the scenes that's quite similar to this. So the specific thing is we can actually try to use this to try and predict something about the cell dynamics. And that's it. And I just want to thank also the students who are now working on this. And this is currently in the archive.
Thank you, Omer. Georg, questions? We have time. Omer took less time than all the other people, so we have time to discuss it more.
You talked a lot about stable states and endpoints. And what does stability mean? Is it really constancy, or are there also oscillations? in your case. Well,
So
how
the way-
could you cover those oscillations we heard about in the last talk?
Yeah, no, that's a great question. So it's about stability and oscillations. So the cell has different components, right? So we have the cell cycle, and obviously oscillates. P53 obviously oscillates. What I'm talking about is not the entire transcriptional network, but sort of like the subset that we think about that has to do with cell identity. And maybe that also oscillates. But the way that we think about this is that this is sort of like stable in the sense that it causes the cell to behave as it does. But the system, it's not in steady state in another way, right? It's because this annealing process is not, it's not a stable state, right? This is a search process that the cell executes. So the cell somehow needs to leave its, so in a way, this sort of like system is outside of steady state in that respect as well. That answered your question.
Hugo?
Thank you for the talk. So my question is, do you have any insight of how aging influences that landscape? So for example, will the tractor basins become shallower? Does one become more dominant?
The answer is no. Maybe you have an idea. But I'll be happy to speak. If anyone has an idea of how something like this could be modulated in aging, I don't know.
Any more questions?
Hi, Omer. Great talk. The question has to do with the temperature term in the annealing process. So I suppose in biology, what's the temperature that drives the differentiation, similar to temperature in annealing?
Yeah, that's a great question. So if you write like a simple toy model, we think about something like histone acetylation, then this process could be driven by something like the deposition of this acetyl terms over the removal. And then we think about something like P300 as something that controls the temperature, or the histone deacetylase is something that reduces it. And we think about the perturbations of these proteins, and they look a lot like we would expect from the model. But I think this is just thinking about only these two aspects is not sufficient. I think that there's multiple levers where the system can control this type of annealing process. Okay, so think about proteins, for example, like MYC. I think MYC is probably a central regulator of this process as well.
Any more questions? 3, 2, 1? No? Then thank you, Omer.